Hello, hello, hello. So my name's Jake Wintermute. I'll start by introducing myself. I come from the United States, but I work 2 countries over in Paris, France, at a place called the Center for Research and Interdisciplinarity. And just as an aside, let me say that this is my first time in Poland, and I'm loving it. I'm having a great time. Yeah. Yeah. I've been here for 2 days, and I've been touring all over Katowice, and one thing I notice is that there's this great energy in the city, there's a lot of excitement, there's a lot of chaos, and everybody who I talk to about Katowice loves Katowice. And it is not like that in Paris. In Paris, nobody likes anything. But that's where I'm from. So this is the Center for Research and Interdisciplinarity. We are a private research organization connected to a public research university. We specialize in 2 things. First, the life sciences, and second, education. So we offer undergraduate, master's, and PhD in the life sciences, the quantitative life sciences, and in education. So my bosses wouldn't let me get away with coming here without doing a little bit of advertising for our institute. We're just now moving into a brand new facility in the Marais neighborhood. It's in the chicest district in Paris. We're going to have maker spaces, we're going to have biohacker spaces, research labs, classrooms, student housing, video production facilities, anything that you want to do an undergraduate, a master's, or a PhD in the life sciences and education. So definitely keep us in mind and maybe come and visit us in Paris or collaborate with us. The talk that I am going to give today has 3 parts: policy, research, and education. And I'm going to argue that all of these 3 things need to come together to bring solutions to some major global public health challenges. And the challenge that I have in mind is tuberculosis. This is a huge global problem, major infectious disease. There's about 2 billion people worldwide who are infected with tuberculosis. That's about 1/3 of the world's population. So approximately speaking, the whole world has this disease. Now, it's not a huge problem in places like Europe or the United States yet, and for that reason, it's a neglected disease. And for that reason, many of our treatments for tuberculosis are very old. They date from the 1950s and 60s. And these treatments are not effective, they're not effective enough, and increasingly they're challenged by the evolution of antibiotic resistance. Why is such a serious problem being treated with such an old solution? Well, mainly, I think this is a policy issue. The problem here is that drug development is very expensive and it's very risky. So here I've laid out all of the different steps in the process of drug development. The whole thing can cost about $2 billion. And every stage in the process has a very high failure rate. Right now, the way that we do this process is in gigantic, vertically integrated pharmaceutical companies. And the pharmaceutical companies are only willing to take these risks and pay these costs to develop drugs that are profitable. Tuberculosis drugs are not profitable. And so that's why places like the CRE and other places that think about open drug discovery and open pharma imagine that it might someday be possible to break this process up and distribute it across many different actors. So you could imagine that some steps of the drug discovery process might be done by academic labs, others by NGOs, others by for-profit organizations. And this is a big policy challenge. There's obviously a lot of legal and regulatory issues that need to be addressed here. I'm not a lawyer, I'm a scientist, and when I think about changing the model of drug discovery, the main challenge that I see is who's going to actually do the work, right? If you take the drug discovery process out of the pharmaceutical companies, who's going to actually do it with their hands? Where does the work come from? I think there's two answers to that question. The first answer is students. These are 3,000 students from the iGEM competition, International Genetically Modified or Genetically Engineered Machines. These students come from 300 teams from universities all over the world. They spend their summer making genetically modified organisms to solve problems. These kids they're very smart, they're very hardworking, they're very motivated, and they definitely have what it takes to discover new drugs if we can just give them the tools to do so. My other answer to the question of who's going to discover the next generation of drugs is biohackers. So it is increasingly true that you don't need to be a student or an academic to do biological research. There are makerspaces, there are community biolabs that are now found all over the world where you can walk in, you can get space, equipment, training that you need to start doing the biological research that you're motivated to do. The global community biolabs are a movement worldwide, including here in Poland, driven by the emergence of this new discipline called synthetic biology. This is a book put together by an iGEM team in Warsaw to describe this new technology of synthetic biology that's coming to Poland and around the world. Synthetic biology is driven by two major trends. The first trend is the decrease in the cost of DNA synthesis. So you're probably familiar with figures like this. This is Moore's law. It shows that the cost of transistors, of computer parts, decreases exponentially over time. And it turns out that biology, DNA synthesis, follows a very similar law. We've arrived at a point in time where it's possible to synthesize a gene from scratch at a cost of about 100 euros. And the price is dropping every year. The other big trend that's driving synthetic biology is the emergence of digital biological science. So DNA sequence is a digital thing. It's a digital beast. We can put it in a computer. I can manipulate it using software. I can cut and paste. I can build a software model to predict how my DNA sequence is going to function. Then I can go online and just type the sequence that I want into a DNA ordering website, and the gene will arrive, and I can put it into a new organism and bring it to life. Moving into the research part of the talk, I'm going to talk about how in my research I try to bring together all of those tools to make new ways of discovering treatments for tuberculosis using synthetic biology. Traditionally speaking, if you want to discover a new drug to kill tuberculosis, you've got two ways to do it. First, you can do a whole cell screen. So you take your grumpy little pathogen bacterium, you put it in a test tube, add your drug that you're looking at, and see if it dies. Right? Now, of course, you can't just do this with one drug. You're going to have to try many thousands of drugs before you discover one that is effective. The other approach is called target-based, and this works on a similar idea. We start with a pathogen, put it in a test tube. Now we're going to grind it up, take out one individual component, an enzyme, a protein, develop a test just for that component, add our drug, and if we can kill this one essential piece of the bacterium, we can infer that this will be a good treatment for the disease. With synthetic biology, it's possible to combine these two approaches. We can engineer safe laboratory model organisms to stand in for dangerous pathogens. Here's how it works. So I've got my grumpy tuberculosis at the top. I find inside a gene that I want to target with my drug. Then I take a happy, safe laboratory bacterium like E. coli, and I find a similar gene. I go in, I delete the gene of interest from E. coli, I replace it with the tuberculosis gene, and now, if I can discover a drug that kills this GMO, this genetically modified E. coli, I can infer that probably the same drug will also kill tuberculosis. And I can do this in a safe way. I never have to actually handle live tuberculosis in order to discover the drugs that kill the tuberculosis. Okay. So this is not a simple technology, right? This is a state-of-the-art genetic engineering, so we have to build DNA circuits in order to control the expression of these genes so that they work in the correct way in the new host. So you can think of this as this is a sort of microchip, right, that we engineer to control the genes that we're interested in studying. And we build these circuits with a plug-and-play technology so that it's easy to remove one gene that we might be trying to target and replace it with a new gene that might also be interesting. And this is particularly important because there's not just one gene in tuberculosis that might give rise to an effective treatment. We estimate, in fact, that there are more than 100 such genes. It's a big genetic engineering project. This is more than we can do in our lab alone, and we know that to be the case. We need to train an army of labs if we want to screen 100 different tuberculosis targets to find the one that is going to be the key to a treatment. And that brings me to the education part of my talk. How are we going to train this army? Where are we going to find 100 labs to discover these treatments of tuberculosis? Well, the good news is you don't need to be a PhD. You don't need to be an expert in tuberculosis biology in order to do the kind of experiments that I've described. And I know that that's true because everything that I've talked about so far is the work of undergraduate teams working in our institute. So we figure it takes about 6 weeks of training to bring someone up to the point where they can do these kind of genetic manipulations, make their own tests for tuberculosis drugs. And there are thousands of people like that in the world who are smart enough and motivated enough to do these kind of experiments if you can just give them the tools and the training. That's why we developed this. This is Synthetic Biology 1. This is a free, open, online course in genetic engineering and DNA design. We had a lot of fun making this course. We were funded in part by the city of Paris, and so we got to travel all over the city, and we get to see the monuments of Paris, and I'm on the Champs-Élysées, and we're talking about genetic engineering, and we make our own GMOs, we talk about DNA editing in software, we work in the lab, we build mathematical models, and we do everything that you need, we teach everything that you need to know to create your own genetically modified, safe bacteria at home. So this is freely available now. You can check it out. You can join us. The vision for this is to take this map, which represents all of the locations worldwide of all of the iGEM teams, where students are learning new approaches to genetic engineering, and this map of all the biohacker spaces that are available in the world where anyone who wants to can come and get involved in biological research, and make them correspond to this map that shows the global scope of the problem of tuberculosis. And with that, I'd say thank you very much.